Okay, let's see if we're recording. Yes, okay, so this is repro4. What I want to do is get into chapter 29. It begins on page 1149. We're just going to do a few pages of this chapter, maybe about 12 pages or so. On pregnancy and childbirth, just get a little bit into it. So gestation divided into trimesters, 3 trimesters, 9 months. Prenatal development terminology, concepsis is what is actually I guess you could say that's the same thing as a zygote. A zygote becomes, remember, a morula. Morula becomes a blastocyst. This happens during the 4 or 5 days that this concepsis goes through the fallopian tubes on its way to the uterus. It implants into the endometrium. It becomes an embryo. Embryo becomes a fetus, and then what's born becomes a neonate. Now, the hormones of pregnancy, we talk about first human chorionic gonadotrophin that's made by a brand new placenta. And we'll see what that does. It does a very important job. And there's also going to be lots of estrogens and lots of progesterone being made during pregnancy. Okay, so there's the wheel of life, so to speak. Fertilization. We'll talk a little about fertilization. And then a zygote going to the cleavage divisions. From zygote, 2-cell stage, 4-cell stage, morula, it really begins at about the 8-cell stage, and then making a blastocyst. And then after implantation, we're going to talk about another really important event in your life, gastrulation, that makes the 3 germ layers. There's a primitive streak made in this disc. And it's just 2 layers, and then gastrulation makes 3 layers: the ectoderm, the endoderm, and the mesoderm. And then after that, we have embryonic periods where we have an embryo becomes a fetus, and then at birth after 9 months. Here is during the weeks after ovulation, implantation at about a week after ovulation. Remember, it takes about 4 or 5 days for the conceptus, what's been ovulated. It takes 4 or 5 days for it to make its way down the fallopian tubes by the cilia that's moving along, and then it bounces around for the uterus for another couple of days. So it's like a week or a little bit more than a week before it actually implants. It forms a primitive placenta, and that placenta makes a hormone called HCG, human chorionic gonadotrophin, and it really goes to high levels. And what this is going to do is maintain the corpus luteum. And the corpus luteum only lasts for 11 or 12 days after ovulation. And so, and we've already had like a week or so, 7, 8, 9 days before implantation occurs. So we got to make this HCG and lots of it. So there's a real time limit here, a deadline. You've got to get this conceptus to this blastocyst and get it implanted, make that new placenta, and secrete this HCG to feed back upon the corpus luteum and stop it from regressing. And then, because it's the source of progesterone, and progesterone maintains the lining of the endometrium. So if the corpus luteum degenerates, the lining degenerates, and this implantation event goes to naught. It will flow out with the menstrual flow. So you have to do that. Because the early placenta, for some reason, can't make progesterone. After about 10 or 12 weeks, after like the first trimester, really, placenta now can make progesterone, and the HCG falls off. You no longer need HCG because now the placenta itself makes its own estrogen. And you can see how estrogen and progesterone become greater and greater as pregnancy progresses, and they're highest just before birth, and then after birth they fall off. So fertilization, sperm migration. Sperm have to migrate from the vagina where they're deposited up through the isthmus of the cervix, and in the meantime, lots of them are killed by the acid conditions of the vagina. They're attacked by dendritic cells. When they get to the top of the uterus, half of them are going to go the wrong way, because 1 month the right ovary will ovulate, and the oocyte will be over here. The next month, the left ovary will make the egg of the month, and it'll be over here. So half of the sperm are going to go the wrong way. And then they have to be capacitated. Capacitation is a thinning of the acrosomal membrane. It has to be thinned, so then when it finally gets to the ovulated object that has the corona radiata, that they hit their head against that, the acrosome splits open, and these digestive enzymes start to digest away these corona radiata cells and a zona pellucida. It takes millions of sperm punching their head against the corona radiata and lots of acrosomes breaking open before it digests it away enough so that one sperm head can get into the secondary oocyte. That's called the acrosomal reaction. Now, capacitation takes 10 hours. So sperm cannot fertilize an oocyte for 10 hours after they've been deposited in the vagina. Then they can have the acrosomal reaction. And then once one sperm head gets into the oocyte, calcium channels are opened up, calcium rushes in and causes this impenetrable membrane to spring up almost instantaneously. This is called the cortical reaction. This can be an impenetrable barrier, and it's going to prevent any other sperm from entering the egg. If another sperm head enters, that's called polyspermia, and it's going to lead to a miscarriage. It's going to really mess things up, and this will be a miscarriage. This thing will not be able to implant. So we have to prevent that by the cortical reaction. Then, after the sperm head gets into the oocyte, it's called the male pronucleus. It has to sit and wait for a while, because the female pronucleus has to go through meiosis too. Remember, it's arrested in metaphase 2. It has to go to interphase 2, telophase 2, kick out the second polar body, and then the male and female nuclei can come together and start going through mitosis. Now we have a zygote, and the zygote goes through mitosis, which are the cleavage divisions, the first mitotic divisions of your life, the cleavage divisions, to make a morula and a blastocyst. And there it is right here. Sperm penetrates corona radiata by the acrosomal reaction. Sperm undergo acrosomal reaction and penetrates zona pellucida. A sperm and oocyte, one sperm gets in there, second polar body is made. Sperm and oocyte plasma membranes fuse. Okay, so as I said, these cells after the first cleavage division are called blastomeres, and they're supposed to become 2 cells, 4 cells. Sometimes these blastomeres separate after the first cleavage division, and they go through the second cleavage division, first cleavage division all over again. This will be identical twins. Sometimes there's something called superfecundation, and that is a woman makes more than one egg in it over the course of the month. Most women just make one, sometimes they make two. And then if they have unprotected sex and there's two eggs present, then different sperm can enter the different eggs, and that is fraternal twins. Identical twins are genetically identical. Fraternal twins are not genetically identical. They're brother and they can be brother and sister. Identical twins have to be the sister, sister, brother, brother. Fraternal twins can be different genders, and they're just as related as regular siblings. They just happen to be sharing a uterus at the same time. Okay, we know what a morula is. The blastocyst, remember, has an embryoblast, which becomes the embryo, and a trophoblast becomes the part of the new placenta. And I just described the difference between identical versus fraternal twins. There's another, and that's called, actually, identical twins are called monozygotic. Fraternal twins are called dizygotic. There's actually a third type of twin, which is incredibly rare, called sesquizygotic, or semi-identical, where two siblings have the same set of maternal alleles and different set of fraternal alleles, which is really bizarre, and nobody knows really how it happens. But I'm not going to get into that. Okay, there is the zygote to all the different cell stages, the morula to early blastocyst and late blastocyst, with the embryoblast, the blastocyst cavity or the blastocele, and the trophoblast. Okay, there are different parts of the trophoblast. I'm not going to get into this too much. Called the syncytial trophoblast, which is the actual part of the trophoblast that makes the placenta, called the syncytial trophoblast. The cytotrophoblast makes part of the yolk sac, I believe. And then remember, this syncytial trophoblast is what secretes the HCG, right? And then, after implantation, embryogenesis begins. Formation of the 3 germ layers. This is gastrulation, very important event in your life. Because initially you will just have 2 germ layers, ectoderm and endoderm, all right? And you have this flat disc that's ectoderm on top, endoderm on the bottom, and the trophoblast, and the cytotrophoblast is below, making the yolk sac. And you have this primitive streak forming in this plate. And what you have is ectodermal cells start to involute in the primitive streak. And this is gastrulation. Well, first here is implantation. Here's the cytotrophoblast, the syncytial trophoblast. The syncytial trophoblast is making the new placenta. And here's the primitive streak. You look at a bird's eye view now, and you're going to have involution. So you have epiblast and hypoblast. Those are going to become ectoderm and ectoderm. And you have involution of the epiblast cells in between the epiblast and the hypoblast. And that becomes mesoderm. And the epiblast becomes ectoderm, the hypoblast becomes endoderm, and what has involuted in between those 2 layers becomes mesoderm. And they make different parts of your body. Ectoderm is nervous system, central nervous system, and skin. Endoderm is your entire gastrointestinal tract, including the accessory organs, and mesoderm is everything in between. Muscles, bones, connective tissue, all what's in between. Starts off as mesoderm. And here's a cross-sectional view of the embryonic disc. And above the ectoderm is the amnion, the amniotic cavity. This is a fluid-filled cavity. Below the endoderm is a yolk sac, and that's going to be forming the digestive tract, the GI tract, and in between, and what's induced is a notochord in the mesoderm from the cells above it, induces this notochord, and the notochord causes the ectoderm cells to rise up and form a hollow tube. And in the very anterior part of the primitive disc, this hollow tube is going to become the brain. The posterior parts of this tube also is going to become the spinal cord. And once that early brain and spinal cord are formed, the notochord disappears and you never see it again. So that's called the embryonic disc. And that's it, folks. So that went up to about page 1159 is gastralization. And then 1160 is the last figure I showed. And then there's always going to be this folding and everything that's going to start making organs, and that's called organogenesis. And we're not going to get into that. Okay, so that's the end of reproduction. We're all finished. So this is going to be repro4. I'll convert this and put this up in YouTube. So we're done. Actually, the video lectures for the semester. We're done. So I'll see you.